so many people, and including our Deputy Director General Research, Dr. Dave Washington. So a very warm welcome to all of you, and I think that we will be having some many more colleagues very soon. So first of all, please join me in welcoming Dr. Ron Phillips from University of Minnesota. And let me give a brief background about Ron, because I know that majority of you are already aware with his name, but some of you who are new to this genomics or the molecular breeding plant genetics, then just to keep you guys updated that Ron has done his BSc and MSc from Purdue University, then he did his PhD from PhD from University of Minnesota, and then he had postdoctoral fellowship at Cornell University. And Ron has been known for many breakthroughs in plant genetics and breeding. And in fact, when I was doing my PhD, so I was telling Ron that as a PhD student, when I started my career in DNA markers, at that time, the first book which I read this was DNA markers in plants from Ron Phillips and I.K. Wassel. So I started basically my career by reading those books. And of course, he is known for several things. One of the important thing is maize oat addition lines, and there are many things you can read on the net or so. So and during his career, he has been guiding several PhD students. I think more than 55 graduate students and about 23 postdoctoral scientists. When I say postdoctoral scientist, many of you may be aware with the name of Oscar Raju, who was our director for the Dryland serials. Oscar did his PhD as well as postdoc with Ron. So, and even Isabel was also in the same group of Ron in Minnesota. And Ron has been on the advisory board or governing board of several organizations and institutes or initiatives that includes International Rice Research Institute, where he served as a governing board member for about 6 years, and many other boards, including Malaysian Oil Palm Research Institute, Donald Dunford Plant Science Center. When Roger Vici was on our board, Ron was on the board of Donald Dunford Center. So those kind of things have been happening, and he was elected at very early age as a member of the National Academy of Sciences of United States, which is considered the most prestigious thing in science. And when I say science, so this includes not only life science, physics, chemistry, etc. So, and he was the elected member of the National Academy of Sciences. And in addition to that, he is having several other awards like Fellow of American Association of Advancement of the Sciences, American Society of Agronomy, Crop Science Society of America, and he has served as a president for Crop Science Society of America in 1990, 2000. Yeah, sorry, in 2000. At that time when you had this meeting in Hamburg, so that was my first international exposure to abroad. So I attended that meeting and I met Ron in that meeting, and after that I kept on meeting. So and right now he is basically helping the World Food Prize Foundation and as well as Norman Borlaug and Borlaug Vici Fellowship. So basically he is contributing a lot in the area of international agriculture. And so we are very much pleased to have Ron with us. So and this is pleasure for us to have you Ron here. And then I invited or requested Ron to give an idea not about the science but something which is very important for international centers, CG centers, and the national programs, and that's the capacity building. So, and I'm very much pleased that you accepted the invitation. And now I would like to invite you to give the seminar. So please join me once again in welcoming Ron.
Well, thanks to everybody for coming, and say hello to people who are watching from satellite areas in India and Africa and so on today. So it's a real privilege to be able to address you. It's been a privilege to be here. I want to publicly thank Rajiv for his invitation and Manju, where she is, for the correspondence I've had in order to plan this trip. It was probably one of the best communicative processes for any trip I've ever had. So it's really a pleasure to be here, and good to see David Hoisington here today as well, an old friend. I was telling somebody that Rajiv told me that he had used our book, DNA Markers in Plants, that I edited along with Basil. And the person I was telling that to said, well, then you can consider him one of your students as well. So maybe you're number 56. I'll accept that. Anyway, what I'd like to do today is to talk quite a bit about genetic diversity, its utilization and detection, also some of the aspects of the need for this, and end up with a few comments about human capacity building. So India certainly has a proud reputation. I've been involved with the World Food Prize, and over the 26 years that this prize has been given, 6 years, an Indian scientist was either the winner or one of the winners of this prize. So you have some really good role models here from India in the area of food production. Starting out with Swaminathan, who, as I understand it, took the $250,000 prize and started the MS Swaminathan Research Foundation and all the good things that have come from that. Kurian I didn't know, but I understand was an economist. Gurdjieff Kush, you'll know probably, is a famous white rice breeder, spending most of his time at Erie. Barwali in terms of the seed industry. Vassal in terms of high lysine, tryptophan, corn, and Gupta from the World Fish Center. So this is a tremendous accomplishment for India scientists, I believe. I don't have very many idols in the world, but I would say Swaminathan's one of them. I admire him so much for everything he's done. In fact, I learned here that maybe the vision for ICRISAT came from him as well, which I didn't know. Tremendous contributions, and the exciting thing is he's still making those contributions, as you know, now a member of Parliament and so on. Nehru, your first Prime Minister, said everything else can wait, but not agriculture. So clearly there's been an acceptance in India that agriculture is extremely important. Borlaug is my other idol. He's standing here in front of Borlaug Hall at the University of Minnesota, where I have my lab and office. We've interacted a lot over the years. You may or may not know that he got all three of his degrees at the University of Minnesota. And Minnesota professors like E.C. Stakeman in plant pathology had a major effect on the international agricultural area of helping to set up CIMMYT and, in fact, helping to prevent Borlaug from getting fired a few times where he didn't want to do the paperwork that was required. So you all know about the world population increase. I've shown here the increase from 1940 to today. I did that because I was born in 1940, and it's just impressive to look at even over my lifetime. It went from 2 billion to 7 billion people. But the way I really like to talk about it is at the rate, and the rate's adding about 1 billion people to the planet every 14 years. That's not very long. So 14 years from now, we could expect that we have to feed another billion people, and it keeps going on without more population control. So the challenge is out there clearly just from that aspect alone. Ambassador Kenneth Quinn, who's president of the World Food Prize Foundation, said the last 50 to 60 years has been the single greatest period of food production and hunger reduction in all human history. That's very impressive. On the other hand, if you look ahead at adding a billion people every 14 years, it means we have a big job ahead of us. And you young people that are here, this is going to be on your shoulders, and it's a very serious matter. So the value of genetics in food production is quite clear. There have been a lot of studies done looking at where does the contribution in productivity, increases in productivity come from. And it turns out, generally speaking, that about 50% of the increase in productivity is due to genetic improvement, improved varieties, and so on. The other 50% due to irrigation, pesticides, management practices, and so on. And mutations, or genetic diversity, has played a major role in this accomplishment. All you have to do is think about the semi-dwarf wheats and rices. So agricultural productivity depends on gene variation. And I would argue that any additional knowledge of gene variation is important in crop improvement. So it's from there that I take my title, that it's genetic diversity is fundamental to crop improvement. So 85 years ago, L.J. Stadler ushered in the era of induced mutations. So that's a long time ago that we knew we could actually generate mutations. Sturtevant said mutations are accidents, but accidents happen. However, nowadays, mutations don't have to be accidents. Zinc finger nucleases and things like that. The picture here is a planting of rice using a purple leaf variety or mutation of rice. And in Japan, they like to create a new figure like this every year. I understand this particular one has Mount Fuji in it. It was 60 years ago this year that Watson and Crick described the model of DNA and its possible mode of reproduction or replication. In fact, I visited the Museum of Science and Technology in Bangalore a few days ago, and they had a display where they said it was on February 28th in that year, 1953, that they actually got the concept of the DNA molecule in mind. So it's just a few days off, and it'll actually be the real 60th anniversary of that very important concept. Now, we've used genomics and molecular genetics in many ways over the years. The use of molecular markers have been very useful in checking the purity of pedigrees and lines and so on. Now it seems like quite old hat kind of work, but very important and very powerful. They enable to look at the degree of inbreeding, looking for the frequency of heterozygosity in our materials and get some ideas as to the amount of inbreeding that occurred. And many other applications, such as describing molecular genetically, a line that's going to be patented or something related to patents. So it can be very important in the patent infringement area. So these are just some of the early uses. Then we went into comparative genomics, looking at one species versus another and finding out that there's relationships there when you have related species and species that aren't really that related. This was the DeVoe scale concentric chromosome model with rice in the middle of that process. 12 chromosomes of rice going clear out to oats. You can see the different species, but if you know where a gene is in rice, you can make some prediction in other species. Not perfect, but very interesting. And in fact, this was, for me personally, I have to admit, this was a surprise to see this kind of relationship. It shouldn't have been, because we know grasses are all related to each other, but since we weren't able to cross them and saw them, this really did come as a surprise to many of us. And then William Basin, several years ago, said, treasure your exceptions. And one of the exceptions right now, I think, is the submergence tolerant rice, discovered in indica rice and transferred to japonica. Erie has done a lot of studies on this and other discoveries from UC Davis and so on. This is a Erie plot where the rice has been submerged for 17 days. It doesn't take very many days, I understand, 3 or 4 to kill rice, but this particular material with the sub-1 or submergence 1 gene can tolerate it. And you can see here in their picture, here's IR64. You can see a few plants came through, but not very many. However, if you have sub-1 over into IR64, you can see the beautiful plot that's there. So in flood-prone areas, which is 75% of the world's rice farms, this is going to have a major impact. In fact, Erie has already produced 10 mega varieties with sub-1A. And they use marker-assisted selection to do this. And by using linked markers, they can transfer this gene into other varieties in 2.5 years. So it's showing nice application right there. They estimate about 100,000 farmers are growing these varieties that they've produced so far. I think it's going to be an extremely important trait. Golden rice, another one that you all know about, produced by Patrikas and Myers. This has been a long time in coming. Been a lot of tests done on it over and over. Now they're using the yellow locust gene from maize to get golden rice. So there's been a slight shift in the approach. Now the request is to actually do human feeding trials and see if it actually works. in the on-ground application. So the estimate is that if that goes well, golden rice might be released around 2014, 2015. So that's something we can look forward to. BT corn, you know about BT, of course. But even though it costs more, the point here is that the farmer gets more back. And this is a Philippine example. But of course, what a lot of the public doesn't know is that by preventing the larvae from crawling through the corn plant all the time, you do not bring in as many fungi and mycotoxins into that material. So you reduce the amount of harmful carcinogens by BT corn. BT cotton, you know very well, particularly here in India, and it's been very highly accepted or adopted here in India. Brinjal, waiting to see what happens, I guess. This is a species where you'd think it should be approved because of the number of sprayings that are done on the eggplant. I've heard on one side of India it's about 20 times, and on the other side about 40 times. But at any rate, there's a lot of sprayings that could be reduced to one or two with this particular trait. We have a lot of genetic diversity, a lot more than we ever dreamed, I think. And corn's a good example of that. The diversity among inbred lines of corn is more than the diversity between chimpanzees and us, which of course we're fairly highly related to anyway. But the tremendous diversity in corn that's being studied now to understand what it all means. Sean Kepler did a study looking at differences relative to the reference genome. He took 500 inbred lines and did RNA sequence on it, and originally found 31,000 unique loci from these lines that were not in the reference genome. With more stringency, he reduced that down to 8,600 loci that are not in the reference genome. So at the DNA level and at the RNA level, we know that there's a lot of diversity out there, and diversity not just in allelic form, but whether it's present or absent. And so these are measurements of genes and gene products that are absent in a line compared to the reference genome. So that's something that was new to us and surely has some meaningful breeding applications. So we kind of come up with the concept of everything being what we might call the pan genome. But out of those lines, there is a certain percent of the loci that seem to be constant. They're there in all these lines. Those genes formulate what's been called a core genome, part of the genome that's there in all the materials. However, in other materials, maybe as many as half, there's a dispensable genome in the sense that the locus may not be there. So we presume it's dispensable. That doesn't mean it's not important, but we are looking at maize now and some other species with the concept of a core genome and the concept of a dispensable genome. So what's the value of this dispensable genome? Well, we don't really know yet, but we can ask the questions, does the dispensable genome affect the phenotype? I presume it does. Are we selecting based on the dispensable genome? In other words, when we make crosses and look at progenies, if everything's got the same core genome, maybe it's not genes in the core genome that we're selecting. Maybe it's the genes in this dispensable genome. And you can ask the question, does compensation of the two dispensable genomes from the two parents affect heterosis? In other words, you've got a certain set of genes missing in parent A, a different set of genes missing in parent B, you bring them together, they can complement each other theoretically and get something better than either parent. So it could have some relationship to heterosis. We'll find out. And you can start asking questions about the basis of selection. For example, are differences in quantitative traits driven by differences in proteins or differences in gene expression? I think generally in the past we've thought about proteins and so on, but not necessarily about the expression. So perhaps there's new breeding methods that should be employed that relate to selecting based on gene expression. You can ask, are traits due to copy number variation? There's a lot of copy number changes in our genomes. And the question is, do those sometimes cause the phenotype? An example here from Science last year indicated that soybean cyst nematode resistance was due to 10 copies of a 310 kb sequence. And this increases expression of a multi-gene segment. And then you can ask, what are the relative contributions of genic and non-genic polymorphisms? A lot of our traits, no doubt, are due to non-genic changes. We worked quite a few years on earliness in maize, and Roberto Tuberosa was part of that, many of you know. Tai-San Kim also was part of that. Anyway, the end result was that it was a non-coding region, about 70 kb from a gene that might be controlled by that non-coding region. So when we're selecting, are we selecting also for non-coding sequences? Just something we wouldn't have thought about a short time ago. Maize has two subgenomes. Maize and sorghum split early on, and then maize became tetraploid, and sorghum remained what we presume is standard diploid. Maize then returned to diploid nature by chromosome fusion, other methods. So the maize genome fractionated and occurred and gave rise to what we're calling maize genome 1 and genome 2. Not me, but Nathan Springer and Pat Schnabel, people like that. So you have two subgenomes, and Springer found that subgenome 1 often has less gene loss than the other subgenome of maize and higher expression. So again, when we select, are we selecting for those more stable parts of the genome and that are highly expressed? So that'd be something interesting to follow up on too. Some people think we ought to have a mutant for every gene. In rice, the estimate's been that it would probably take between 180,000 and 460,000 lines in order to have a mutation in every gene. So people are working toward that goal, also in Arabidopsis and so on. And in rice, there's 200,000 mutant lines that are available to you right now. So it does mean you may not have a mutant in every gene, mutation in every gene, but have a mutation in many genes that you might be interested in. A lot of good materials being developed. The TOS17 transposable element system in rice has generated about 50,000 lines. And these elements have been moved around in different loci and have many sites that are marked due to this repeated element. So when did genomics begin? Well, you might argue it began 1977, 36 years ago, by the sequencing of Phi-X174 DNA, which cost a lot of money at that time, but a very small genome. But that perhaps is the first case where you have a complete sequence of an organism. And then, of course, over the years, we've sequenced many species. This is an early list, actually, but I show it mainly just to indicate the size. Arabidopsis thaliana was first sequenced 130 million base pairs, which illustrates why we have to have computers and softwares to analyze these data. This Arabidopsis was chosen because it goes from seed to seed in about 60 days and has one of the lowest amount of DNA of any plant species. Rice was the second one sequenced, has 430 million base pairs. This was chosen because it is a cereal with probably the lowest amount of DNA in cereals. And then you can see many other species, including maize down here, that has 25 or 2,600 megabases, about the size of the DNA in each of our cells. So as of last September, this was a list of species with sequences or near sequence genomes, and you can see it's quite extensive. A little too early, I guess, to have the chickpea genome on there, but it'll be on there next time around. Beg your pardon? Pigeon pea is up there? Oh, I'd forgotten that. Aha. So, at any rate, it's expanding tremendously. A lot of resequencing going on where you have a reference genome in a species and then you're sequencing others, at least for the gene space region, and making comparisons. The cost of the technology is going down tremendously, and will continue to go down, I'm sure. The current cost of sequencing a megabase of DNA is about 10 cents. In 2001, the cost was $10,000 for that same amount of DNA. So now the cost of phenotyping is more of an issue than the cost of genotyping. And the estimate's been made that the cost of genotyping is perhaps a third of what it costs to phenotype or test the line. So phenotypic evaluation is now the game changer. And so people are starting to think more seriously, as well as you are, about high throughput methods and accurate methods to phenotype your materials. In 2009, the CGIAR had a meeting in the Netherlands, and I was asked to talk. Actually, talked there as well. And I made the bold proposal, I guess, at that time that the cost of sequencing was coming down to the point where we could consider sequencing the entire set of accessions in the Erie rice gene bank. That's over 100,000 lines. And today, that's clearly possible. Since then, Erie has joined up with the Beijing Genome Institute and sequenced 3,000 lines of rice. It took them about a month to do that. It took them about 12 months to assemble all those sequences, but at any rate, within a year's time, they had information on 3,000 rice accessions. My understanding is that they're going for 10,000 now. And if the 10,000 indicates some real value and real applications of that information, they're going to try to do the whole gene bank. So people are thinking in those kinds of terms now. Not only sequencing a reference genome, but sequencing many others. And I was impressed when the chickpea paper came out that they not only had a reference, but they had 90 accessions also sequenced. So this is becoming the norm to do many, many accessions of a particular species. Sorghum, there's supposed to be 100 complete genomes soon, as I understand it. Wheat's considering doing, Simmet is considering sequencing 150,000 strains, according to Ravi Singh last summer. Of course, we have the 1001 Genomes Project in Arabidopsis. Oryza rufipogon, one of the ancestors of rice, 446 accessions been sequenced from various geographic regions. And then the BGI's got a project they call 1,000 Plant and Animal Genome Project. Somebody asked them how they're going to choose those, and they said, well, we'll just take those that are tasty. So good a criteria as any, I guess. But we know, of course, wild species have a lot to offer, and it's a largely untapped reservoir of genetic diversity that I'm sure genomics and future methods will allow us to utilize that material way more than we have so far. And then, of course, there's all kinds of databases, and I just thought it'd be fun to put together some of the rice ones here. All kinds of expression, clear down to cis-acting elements and so on. There's many, many more. But anyway, it's a wonderful thing that comprehensive databases for different species are being developed and useful, and you can access them very readily. Then, of course, we have the next generation sequencing. In 2012, this last year, the estimate was that throughput can be equivalent to sequencing one human genome at 1x coverage per hour per instrument. In fact, I even read something the other day where they were talking about sequencing the genome of unborn children, the fetus. So when they're born, they know the sequence, and if we know anything about diseases and so on, that kind of information will be there when the infant's born. So we have to think outside the box, particularly in our crop science area, and assume that the technology is going to keep moving fast. A lot of times when you discuss with people, the assumption is, well, we'll consider our today's technology and figure out what might happen. Well, that just doesn't work in the area of genomics. SNPs, single nucleotide polymorphisms, are very numerous. Again, for me, quite a surprise. In fact, I understand in the chickpea, it's like 4 million have been identified among all those materials. It's just fantastic. So marker availability is there to do, seems like most anything we might want to do. In rice, there's studies using at least 160,000 SNPs that came from 20 diverse varieties, land races. Chickpea, you know about, study by Gower recently reported using 76,000 SNPs, brachypodium 2.5 million. A fluidine system allows 9,200 SNPs to be assessed in 4 hours. In fact, that 4 hours, there's only 20 minutes of hands-on work. All the rest is done by the instrument. And it comes down to about 7 cents per genotype. So it's unbelievable. But SNPs don't even reveal the entire amount of variation that's there. There's a lot of structural variation as well. Indels, insertions, deletions, inversions, translocations, copy number variants that we talked about are all there as well, in addition to just single nucleotide polymorphisms. And in many cases can be used to our advantage. This is a slide Bob Stupar, one of my colleagues, gave me about soybeans. It shows the 20 chromosomes of glycine max. The blue lines above the line represent duplications, and the red lines represent deletions. And so you can see that it's very, very common across all chromosomes. Then we have DNA methylation, again, that can affect expression and may well be as we select for stress tolerance or whatever, we may be selecting for changes in methylation. So we have a lot to learn there. Rob Martinson recently said one genome, many epigenomes. So there's a lot of variation out there, in addition to the sequence information. That's no doubt going to be very important. We have forward and reverse genetics. I won't go through all these, but all kinds of elements that can move around and tag different genes. Then we have what I've called de novo variation. That is variation that occurs in the progeny that was not there in the parents. And so there's many mechanisms by which this can occur: intergenic recombination, unequal crossing over, naturally occurring point mutations, of course, transposal elements, DNA methylation, paramutation, gene amplification. All kinds of mechanisms can give something different in the progeny that you really didn't see in the parents. So what's this mean? Does it mean that we can go into homozygous inbred lines and make selections because of these mechanisms that can occur in the progeny materials? Something to think about. And then, of course, gene additions, adding genes to species that were not there, what I'm calling biotech crops here. And it shows the developing world here, coming up since 1996. That's when it was first commercialized. This is the industrial world, and this is the total. And you can see in 2011, these figures matched each other. And I just happened to get an email from Clive James this morning, and he said that the adoption of biotech crops in the developing world has now exceeded the developed world as of the last data set. And many countries participating in this, and I just show this. India over here is number 4. So 82% of the cotton is transgenic, biotech, 75% of the soybeans, 22% of maize, 26% of canola. I'm always kind of interested in the maize because they use tissue culture regeneration, which our lab was the first to do. This just shows the different crops and how the adoption has gone on. This is canola here, cotton, maize, soybeans. You see soybeans really took off. So let me give you one other example from some of our work, what we call uniparental chromosome loss. Turned out that Laurie and Bennett in the UK crossed wheat by maize and rescued the embryo after a couple of weeks' time and got plantlets, and those plantlets were all haploid wheat. So what happened was that the genome of maize and the genome of wheat came together to make a hybrid zygote, but in the early stages of embryo development, the maize chromosomes were eliminated, and you were left with just a haploid set of oat chromosomes, which was considered a real breakthrough as another method of producing haploids. So Howard Rhines, who was an oat geneticist, and I teamed up in the department and started working on this. he was interested in haploid oats, and he'd been transplanting anthers of oats and probably transplanted 100,000 anthers and got one haploid plant. So it wasn't very efficient and needed a better method. So we started crossing maize and oat together and then rescuing the embryo after a couple of weeks' time here and grew those plants out. And the plants were haploid of oat, which meant they had 21 chromosomes. However, as we went further, and with the help of Oscar Lozarazu, we found something else. We found that 2/3 of the time, after you make that hybrid, there's a complete elimination of the maize chromosomes, giving you an oat haploid, which has 21 chromosomes. However, about 1/3 of the time, not all the maize chromosomes are eliminated, and you're left behind with one or more maize chromosomes. And we've gotten all the way from 1 to 10 left behind, but it's only when you have 1 or 2 that the plants will produce any seed. After that, the plants aren't very happy campers. So it definitely has an effect. But you can select in lines with one chromosome added to the oat genome, one chromosome of maize added to the oat genome. This is a haploid-diploid comparison of oats. You can see the haploid is a very nice plant in oat. This shows a haploid compared to a haploid of 2 maize chromosomes. And you can see it does stunt the plant and so on, but it's obviously a growing plant. We also, and this was actually Oscar's work, doing genomic in situ hybridization using labeled maize DNA and trying to see what these chromosomes were. And you can see the 2 maize chromosomes just stood out like a light bulb. In fact, when Oscar saw it before doing a Skish analysis, he thought these were fragmented oat chromosomes, but they look like maize, and so we did this, and sure enough, it showed up. And it's another example of how the technology's advanced. When I was in grad school, we were crossing maize and trypsin. Dave, you remember all that? Spent their whole lifetime career trying to prove what they had, right? Whereas with this kind of technology, in a matter of seconds, you can see what's going on. So this just is a table showing the addition lines where we've got an oat, maize chromosome added to oat. So in the Seneca 60 background of maize, which is what they used in England, we got lines that for all 10 chromosomes, one at a time, added to the oat background. Chromosome 10 did not transmit as a whole chromosome, but tended to break and would transmit the long arm, but would not, or short arm, I guess it was, but would not to transmit the whole chromosome. And then we did some work with B73 and MO17. We didn't get a complete set, but between the two genotypes, we have a complete set. Genetic background does make a difference. Then we took those materials and we irradiated them with cesium and generated breakage of the maize chromosomes there. Probably broke the oat chromosomes too, but oats is an allohexaploid and can tolerate a lot of changes. But anyway, the pluses indicate where we used 40 to 50 markers. The plus indicates that marker was there. Over here, then, this is a deleted piece of the corn chromosome. Here's another big deletion. Many of these are translocations of the part of the maize chromosome over to oats, so it will transmit. But you can see the wide variation. We've got about 600 lines like this. So it means that we have available then oat lines that have various pieces of maize chromosome. Phenotypically, again, it depends on the background, but this is chromosome 3 of maize added to oat compared to normal. You get this hooked panicle and you get messed up ligules in the upper nodes, which actually is the same kind of phenotype of ligulus 3 mutation in maize. And we've shown that this is due to ectopic expression of that gene. Again, in certain backgrounds, chromosome 6 gives what we call lesion mimic phenotype. There's no disease here, but it looks like there is. It's a genetic change due to having maize chromosome 6 added to the oat genome. So I wanted to just give one example here. We're crossing oat and maize together, which means we're crossing a C3 and a C4 species. So it'd be interesting to follow up on this material to see if you could transfer aspects of C4 photosynthesis to oat. And we've done just a little bit of work and published one paper on it, but it's waiting there for you guys to work on, or anybody that wants to. We know, for example, from other mapping that the PEP carboxylase gene is on chromosome 9 of maize, and the pyruvate orthophosphate dikinase, PPDK gene is on chromosome 6. And then we also see some bundle sheath abnormalities when we have chromosome 3 added. This just shows the PCR amplification using the PEP carboxylase. Of course, it shows up in the original maize line. But these are DNAs from each of the addition lines with chromosomes 1 through 10 added one at a time. And you can see that when you have DNA from OMA 9, it amplifies. So the gene is there on 9 as we expected. PPDK, we expected on chromosome 6. It turned out it was only DNA from that particular addition line that amplified. So things seem to be working right, and be interesting to follow up on that. Well, I want to end with just a few comments about human capacity building. Certainly the fact that 6 out of 26 years an Indian scientist won the World Food Prize they have to serve as role models. And if I were you, I would certainly play on that. It's an important accomplishment. And then there's other advanced training programs, and I want to just mention the Monsanto Beecher Borlaug International Scholars Program, which I'm on the panel for. This will be our 5th year, and last year, the original program, $2 million a year, $10 million over 5. And we've given out, up until this year, 52 awards, and 14 of those went to students from India. So again, the highest proportion of any country. So this program has been extended for 3 years, not at $2 million a year, but at $1 million a year. So I presume, so we have our meeting in just a month or so for this year. So we'll have another 12 or 14 students funded over the 52. But then we're going to have 3 more years at $1 million. So we'll be adding 5, 6, 7 students a year. This requires a collaboration between a developed country lab and a developing country lab. So with the student being kind of the liaison between the two. So you have to work out that collaboration in advance. Student has to be willing to work in the public sector when he or she gets done. And spend at least half a year in the other location. So they could be located in the developed lab or the developing country lab, but they need to spend time. And the advisor gets some money too. The scholarship to the student, the stipend, is set at a higher rate than is normal in their department, wherever they come from, just to make it seem like a more prestigious award. So we funded 52 from 21 countries at this point. Another program I'd like to promote, I guess, is short-term exposure to international agriculture. When I was on the board of trustees for Erie, I was always very impressed with the discussions that I was part of or overheard. It's a very serious business among those scientists, and that's what I've found here at ICRSAT as well. I didn't hear any flippant remarks, and I thought, wow, what a wonderful atmosphere it would be for our students to be able to get exposed to that. Well, it turns out that our students are having to learn so much technology and so on that they really don't want to leave for a year or 2 years to do a thesis, but they'd love to go for 2 or 3 months and be in a lab and try to soak up what they can. And so we developed a memorandum of understanding with Erie between the University of Minnesota, and it's been just great. Students who go learn a lot. I know this isn't the approach that a lot of people like to take, but my thought is that the current students, a lot of them will and are taking up these kinds of short-term experiences. And then later, when there's a job available at an international agriculture research center, they probably will be much more likely to apply because they've seen that it's serious business, they've seen there's good lab, good scientists, good opportunities there. So I would, and I know you're doing some of this, but I would just encourage this kind of approach. So in summary, let me say that the inherent gene variation is more prevalent than we ever expected before. The new technologies are and will continue to expand the boundaries of gene variation as we learn more and more. And it starts to raise all kinds of questions, and you can make your own list. Some of them would involve, are we selecting based on the dispensable genome? Are quantitative traits driven by differences in proteins or differences in gene expression? Probably the latter. Is selection often due to copy number variation? Can be. Are subgenomes of different value? Can we capture de novo variation in our materials? The biotech crops show the value of adding certain genes to our crops, and human capacity building certainly is essential. So genetic diversity, human capacity building goes hand in hand as being fundamental to crop improvement. So there's this balance between genetic diversity and crop improvement that we have to continue to keep in mind. So the question is, can we even imagine the future of crop genomics? And all I can say is, I don't think so. I don't have to think back very long, certainly not more than 15 years or so, and I would have never imagined we'd be where we are today at that time. So let me end with a quote from Einstein, who says, I'm enough of an artist to draw freely upon my imagination. Imagination is more important than knowledge. Knowledge is limited, but imagination encircles the world. So I'd encourage you to use your imagination, put your feet up on the desk and think about what you might do in the areas that's of interest to you, and enjoy the best job in the world. Thank you.
This was really very wonderful and impressive presentation from Ron, and he took us through a journey from Stadler's time from the mutations to the genome sequencing, where you can analyze or you can understand the variation at single nucleotide level. Again, as Ron mentioned and concluded in his last slide, that imagination, we need to be really imaginatory. And as we know, and at least when I did my PhD, I was starting the PhD, I knew that from the literature at the time when in crops, when genomics started, people started to develop the RFLP maps in the different crops like University of Minnesota, and when Dave was at Missouri, so these guys were doing maize RFLPs, someone is wheat RFLPs, barley RFLPs. That was the era of RFLP map. Nobody thought at that time point that after maybe 20 years later or so, each crop will be having the genome sequence, and not only the genome sequence, you will be having that resequencing of hundreds of the thousands of lines. So this is the beauty of genomics. And next thing is, which I would like to once again recognize contribution of Ron in international agriculture, as he was guiding IRRI through its governing board member, and I believe that even for Summit, and when Dave started, or so at that time point, Ron helped Summit to establish their biotechnology center, applied biotechnology center. And of course, through the World Food Prize from this Borlaug Fellowship, and he's contributing a lot. And now we are also getting his advice and suggestions for our Center of Excellence in Genomics, and then how, and as you know, that we have this critical focus area of molecular breeding, how to extend it, the CFA molecular breeding. So I think that we had wonderful presentations, but in case if you are having some, or you would like to have some suggestions or some advice, some blessings of Ron in your career, you would like to know something, because majority of the people there young, and majority of us are starting our career. So you can ask, or we can have 5-10 minutes for those discussions. And I hope that Ron will be more than happy to give those kind of suggestions and advices. People are shy. You would like to ask something, Abby? And and you should also mention that in Australia you were working on wheat, and now she is working on chickpea. So it's also transition from the major crops to so-called less studied crops. So this is wonderful thing. So from government of Australia, for instance, yeah.
that's great. I just wanted to comment that Borlaug himself believed a lot in training. As you know, he trained hundreds of students, some young, some not so young, I guess, but to go back to their country, go back to their country and work. So Borlaug believed in science and believed in education, training. He believed in being part of the policy making and politics involved. You know, when he had his tweets that he wanted to send to India and Pakistan, he had quite a time. And most of us would have given up, I think. In fact, I understand he had truckloads of wheat seed to go to India and had a hard time getting through the Mexican border, U.S.-Mexican border. They finally got through. I think it took a couple of days. Um, then he found, when he got to Los Angeles trying to reach the port, that the Watts riot was going on. I don't know whether you remember that or not, but fires and fighting and everything, and that hung him up. And the ship was going to leave without a seed, and he got a hold of, I think, of the president of the company, and finally got him to wait for his seed and got it sent. So, very determined person. But anyway, the point is, he believed in science. He was quite supportive of biotechnology after a little bit of time. Believed in training very much, and believed in being part of the policy making process.
Anything else? Dave, you would like to add something?
Always ask Ron an interesting question. Um, you've been in academics all of your career, um, and I think that, as you mentioned, I mean, the CGIAR, I think some of our best work has been in really basic science. But you've also probably experienced in your association with the CGIAR that we're being pressured more and more to have impacts in farmers' fields, which kind of gives us in this dilemma of where do we put our efforts? Trying to get products out, grown, and have impacts, because like you say, population is growing, many other things are challenging us all, versus what we really would like to do, which is sit back, put our feet up on the desk, and imagine. How do you see that balance? How do you see us resolving that?
Well, I don't want you to get lazy and go to sleep. No, that's a good question. I'm kind of a person that spans the basic to the applied, and so I believe there has to be a balance. I think the basic information gets us excited, gets us knowledgeable, which we can apply that knowledge in ways a lot of times we don't even realize we're applying, based on knowing something about how complicated the genetics is, what the genotype by environment interaction is, and all those kinds of things. And with the new tools, can make some advancements like the sub-1 gene in rice, being able to transfer that in 2.5 years. So to me, there needs to be a balance at the research institutes between learning basic knowledge and applying it, but it can't be all one or the other, in my opinion. I think if you look at the really strong centers that we have, most of them have been very good in basic research. In my experience with Erie, you've got a lot of good research that's gone on there, and yet, of course, some real applications, and originally in just traditional breeding, but now in molecular breeding. And they're looking ahead to many new ideas, such as C4 rice and climate change kinds of activities. Um, so I think that two have to go together. And I know you get pushed hard. Even when I was interacting with Cinnamon originally, I think you were getting pushed by UNDP to do some more upstream research at that time, and decided to do molecular genetics and RFLPs, and Dave came and had that program. But at any rate, I think it keeps us stimulated to do the basic research and understand it. And when we do apply it, then I think we can do so in a more consistent and effective manner. So I would call for a balance. Well, I think it depends on where the funding comes from in part, but I think it's reasonable to expect that people will go back and help their home country, and that should be encouraged. Um, there's a program that I actually suggested and been the director of for 26 years called Project AgGrab, where we bring students from a developing country to study production agriculture at the advanced level. They'd all been from Africa up until this year, and now we have a student from Nagaland, India. Uh, but we ask these students to go out and talk to people, different churches. It's a church-related activity. And by the time they've given their talks, saying, I'm here to learn to take back to my country, they go back to their country. So I think, encouraging students to express their reasons as to why they're here helps in itself in them not veering from the idea of going back. You have quite a guilt trip if you don't. I mean, it's going to happen once in a while, but I think it's important to go back. And nowadays, there's a lot of opportunity, a lot of funding emphasis in Africa and other developing countries that we didn't have before. Centers like ICRSAT and some tremendous opportunities. So I think the reasons for not going back are fewer than it used to be. And so you don't want to drain the smartest people away from a country where they can go back and make a major contribution. But the government has to be supportive, and that's happening. Dave knows a lot better than I do, I'm sure, but many of the African countries are arguing to give a certain percent of their GDP toward agriculture now. You're beginning to realize that, and if you look at some of the studies that Phil Party's done on economics and funding of research in the international area, how that went down so tremendously, and now it's improving. Kind of took the Bill and Melinda Gates Foundation to wake us up, I think. But anyway, times are changing in a positive way, I think. And getting more and more trained people back in home countries who can then train and help new people to come back and solve is very important. So, I think you have to try to make the system work in a way that people want to go back. Okay, comment about the communication of science to the public. It's a real challenge. It's a challenge to, first of all, be well informed as to what you say, and it's a challenge to try to say it in a way that the public understands it. It's partly a literacy problem starting from very early on. So I think in our schools, very early grades, people need to be into that. Rajiv's daughter's talking about the chickpea genome last night. Any rate, it is a challenge, and I think you have to recognize that, generally speaking, it's not the science that they're concerned about, but some inner feeling about altering the food supply, an inner feeling about having multinational corporations controlling our food supply because of patents. And I think things like you're trying to put together here, a public consortium relative to transgenics is a good thing to do. I think in the States we have a hard time with that because we take so many funds from companies that even though you say you're public, they'll argue with you that you're tainted by private monies. But anyway, I think the problem, one of the problems is just getting the forum. If someone on the anti-side, makes their remarks, it hits the paper, it hits TV. But if you're positive, it usually doesn't make it. And part of the other problem, in my opinion, has to do with one type of agriculture pitted against another. And if you look at a lot of in the United States, if you look at a lot of the anti-comments, it comes from the organic area. As much as organic may have its place, a lot of it in the States is trying to argue against biotechnology in favor of organics, which in fact with the E. coli 0157H7 organism, organics are not totally safe either. So I don't know how you get around it. The public certainly has their opinion. Europe hasn't helped because they're banning a lot of the biotech work, even though their own studies indicate that it's safe and has value. So I wish I were better at communicating with the public, as I'm sure we all do. But I think we have to do it and try to do it in a fair way. We can call a spade a spade. But again, it's hard to make the headlines with a positive statement.
So I think that we had wonderful discussions as well, and just like to close the session with a light discussion of just to elaborate about chickpea genome by my 4-year-old daughter. So, and this is just an example that, well, when you talk, and we never realize that I talk about genome at home so frequently that on one Sunday, and generally at our home on Sunday morning, my wife, she prepares a chickpea curry called chole. So my 4-year-old daughter asked, Mom, what you are preparing? Then she says, chickpea. Then she says, chickpea genome. So this is the beauty that when you keep on talking, so maybe as a scientist, as you say, that maybe we need to be more communicator to the people, then people will start to learn that, well, what we are talking. Anyway, so thanks once again, Ron. So please join me in thanking Ron once again for this wonderful presentation. And now I would like to invite you for a cup of tea and coffee, and this we will go in that.